We are doing something today that feels a little bit like stripping the paint off the walls of a house to see what is actually holding the roof up.
Right. Down to the studs.
Exactly. Usually, we look at a specific trend or a news event or some kind of medical breakthrough. But today,
Mhm.
we are going down to the absolute bedrock.
We are going to the basement, the foundation. This is, in a way, the first episode of this whole series on pathology, Patho 1.
We are laying the intellectual groundwork for, the entire study of pathology. And I think for a lot of people, when they hear that word pathology, they think of a morgue, they think of CSI.
Yeah, forensics.
Autopsies.
But the source material you've brought today, it suggests that pathology is actually the study of life. Just life under pressure.
That is the perfect way to frame it. If you really want to understand health, you actually have to understand disease. You can't really understand how an engine works until you hear it making a weird noise.
That's a great analogy. And today, we are traversing the landscape of the cell. And I don't mean the diagram you had in high school biology with the mitochondria drawn as a little jelly bean.
Right. Not the placemat version.
No.
Though.
We're looking at the cell as the fundamental unit of suffering and also the fundamental unit of survival.
And to guide us through this, we are synthesizing two absolute titans of the field. These are the Bibles. You walk into a medical school library anywhere on Earth, you find these two books.
You do.
We have Robbins' and Cochrane Pathologic Basis of Disease, and we have Underwood's Pathology.
Right. And they are two very different beasts, which is why bringing them together is so interesting. Robbins, as you said, is the encyclopedia. It's the mechanics manual.
It's exhaustive.
It gives you the breadth, the unifying framework, and, the crushing molecular detail. If you want to know the specific protein pathway that goes wrong in some rare cancer, Robbins has it.
It's the how.
Exactly. It's the deep mechanism.
Right.
But then you have Underwood, and Underwood is, well, it's more like the philosopher of the two. It provides the clarity and the grounded reasoning. Underwood is constantly stepping back and asking why. Why is the body reacting this way? It views disease not just as a broken part, but as a response to injury and stress.
So before we fully jump in, we really need to issue a clear disclaimer here. We are doing a synthesis. We are creating a conceptual map, trying to connect the dots between these two massive texts. But just listening to this conversation, that does not replace reading the books, does it?
Absolutely not. And we have to be really clear about that. You cannot become a pathologist by listening to a deep dive. The goal here is to give you the mental scaffolding.
Right.
We want to prime your brain so that when you do open those chapters, when you read about, say, receptor-mediated endocytosis, you aren't just memorizing a term. You're fitting it into a story, a framework that we started to build today.
A story that starts with a little history lesson that really, it blew my mind. I think most of us, if we're being honest, still kind of think of disease as something that just happens to us. Bad luck, a curse.
Right, yeah.
And the intro material points out that for centuries, that was literally the medical consensus.
It was. You had the theory of the humors. You had spontaneous generation. The whole idea was that disease was like an imbalance of fluids or just some mystic cloud that descended on a person. It was very abstract.
And then comes Rudolf Virchow,
the father of modern pathology, mid-19th century, and he just changes the entire game. He puts forward this concept of cellular pathology. And it sounds so simple to us now, but at the time it was a complete revolution.
What was the core idea?
He said, disease originates from specific perturbations in molecules and cells.
Meaning there is no such thing as a sick person in the abstract.
Correct. There is no sick person, only sick cells. If you have a pain in your chest, it's not some vague heart trouble. It is a specific group of cells in your myocardium that are screaming because they can't get oxygen to run their pumps.
It's so much more specific.
It moved medicine from mysticism to mechanism.
And that shift from magic to mechanism, that's what allows us to actually treat things. If it's magic, you pray. If it's a mechanism, you can intervene. You can fix it.
Precisely.
Yeah.
And that leads us to the core mission of this deep dive today. We have 4 big takeaways we really want to land. If you remember nothing else, remember these 4 things.
Okay.
First, disease is fundamentally a failure of adaptation.
Adaptation versus failure. We're going to spend a lot of time on that.
Second, the cell is an organized system, not just a bag of jumbled parts.
Yeah, it's not a soup.
It's not a soup.
Yeah.
Third, what a cell looks like, its structure, its morphology, is a direct reflection of whether it's working or failing. And finally, you cannot spot the abnormal if you don't deeply intuitively understand the normal.
Let's start right there then. Section 1, defining disease. It feels like such a basic question, what is disease? We know it when we feel it, but Underwood gets really philosophical about this.
Underwood loves to challenge the premise, and he starts by asking, what is normal? In our daily life, we think normal is a fixed thing. Like 98.6 degrees Fahrenheit is the normal body temperature.
The gold standard.
But in pathology, and in medicine generally, normal is statistical.
What's a bell curve?
It is. When you get a blood test back, and the report says your sodium level is normal, all that means is that your number falls within 2 standard deviations of the mean of the population that they tested.
So being abnormal just means you are rare, statistically speaking.
In a strict statistical sense, yes. And this is where it gets really tricky. Underwood draws a very sharp line between what is usual and what is normal. And this is a distinction that honestly trips up a lot of medical students.
Okay, help me visualize that. Give me an example of something that is usual but not normal.
Okay, the classic example is atheroma. These are the fatty plaques that build up in your arteries, the
Right.
things that lead to heart attacks and strokes.
Right.
Now, if we were to autopsy a random sampling of, say, people over 80 years old, finding atheroma in their arteries would be extremely common. It is usual.
You'd expect to see it.
You would absolutely expect to see it. But is it normal? Is it a sign of health? No, of course not. It is still a pathological process. It is still a disease state that restricts blood flow and can kill you.
So just because everyone has it doesn't make it good.
Exactly. Just because it's usual doesn't make it normal in the biological or, the healthy sense.
And contrast that with a 15-year-old.
Right. If a 15-year-old has that same level of atheroma in their arteries, it is both abnormal and unusual.
So context is everything.
Context is everything, which bridges us to that first big takeaway. Disease as a failure of adaptation. I love this section because it makes the body feel so dynamic. It's not a statue, it's a boxer in a ring.
That's a great analogy.
And Underwood frames health as successful adaptation. I mean,
Yeah.
think about it. Our environment is constantly trying to kill us,
It really is.
quite literally.
Yeah.
You have gravity, radiation, temperature fluctuations, pathogens, bacteria, viruses. They're all punching at us constantly. Health is the body's ability to slip the punch or, take the hit and keep standing.
The example that made this click for me was the suntan.
It's the perfect classic example. UV radiation from the sun hits your skin.
Mm-hmm.
That is an injury. It's a physical stressor that can damage DNA.
Right.
So how does the body respond? It doesn't just sit there and take it. The melanocytes, the pigment cells in your skin, they ramp up production of melanin,
They make more pigment.
and that melanin arranges itself into a little cap, a little umbrella, over the nucleus of the cell to shield the precious DNA from further radiation.
So a tan isn't just cosmetic, it's a callus, it's a shield.
It is a shield. That is successful adaptation. You adapted to the stress. Disease occurs when that adaptive capacity is exceeded. You stay in the sun too long, the melanin isn't enough, the DNA gets fried anyway, the cell burns, and potentially you trigger a mutation that leads to melanoma. Disease is the moment the boxer gets overwhelmed and knocked out.
This leads perfectly into the section on Darwinian medicine. This was, I have to say, arguably my favorite part of the reading. It just flips the script on how we think about symptoms.
It really challenges our ego. We tend to think of our bodies as these perfectly designed machines, designed for our personal happiness and longevity. We think we are built to live to be 100 and be pain-free.
And Darwin says, not so fast.
Darwin says, I don't care if you're happy. I don't care if you're in pain. I only care if you survive long enough to reproduce.
That's it. That's the whole game.
From an evolutionary perspective, our bodies are designed for reproductive success. That's the only metric that matters in the grand scheme of things.
So how does that change how we view a symptom like a fever? Because when I have a fever, I feel miserable. My first instinct is to take a Tylenol, get rid of it.
Exactly. We treat the fever as the disease. But Darwinian medicine asks a different question. Why did we evolve the capacity to get a fever? It consumes a huge amount of energy to heat the body up. It's metabolically expensive. If it were useless, evolution would have discarded it millions of years ago.
So it's not a bug, it's a feature. It's a weapon.
It's a defense mechanism. Many bacteria and viruses have a very, very narrow temperature range where they can replicate effectively. By cranking up the body's thermostat, we're literally trying to cook the invader out.
Wow.
And we're also speeding up the metabolic rate of our own immune cells, making them faster and more effective.
So by taking the Tylenol.
By suppressing the fever immediately, just to feel comfortable, you might actually be disarming your own defense system. You could be prolonging the infection. You are trading comfort for efficacy.
That is wild. It makes you rethink the entire pharmacy aisle. They mention obesity in this context too, which I think is so relevant right now.
It is maybe the most pressing example of this. Why is obesity so prevalent? Is it just that modern humans are lazy? Is it a willpower disease?
That's the narrative, often.
Darwinian medicine suggests it's actually a success story gone wrong. It's a legacy of our Stone Age bodies living in a space age world.
Because for 99% of human history, there was no fast food.
For almost all of human history, calories were scarce and unpredictable. Starvation was the main threat. So the bodies that were best at grabbing every single available calorie and storing it efficiently as fat, those were the ones that survived the winter. They were the genetic winners.
We are the descendants of the best fat storers.
Precisely. We have Stone Age genes in an Uber Eats world. Our adaptation, our incredible ability to store energy efficiently, has become a liability because the environment changed faster than our genes could.
Okay, so before we get into the nuts and bolts of the cell itself, the source material says we have to clean up our language. Pathology has a very specific lexicon. If we don't get these terms right now, we're going to be completely lost later on.
Yes, accuracy here is absolutely critical. In common speech, we use words like cause and symptom very loosely. In pathology, they are rigid, well-defined concepts.
Let's hit the core four. The first one, etiology.
Etiology is the why. It is the cause. But Underwood makes a really crucial distinction here between the agent and the cause.
Wait, I thought the agent was the cause. If I get the flu virus is the cause, right?
Is it? Let's use tuberculosis as our example. The agent is the bacteria, Mycobacterium tuberculosis. You cannot have TB without that bug. It is a necessary component.
Okay.
But is it sufficient?
Meaning, does everyone who gets exposed to the bacteria get sick?
Exactly. And the answer is a resounding no. Many, many people are exposed to TB, their immune systems handle it, and they never develop the active disease. So what is the true cause? The cause might be the poverty, the malnutrition, or the overcrowding that weakened the person's immune system and allowed the agent to take hold.
That's a massive distinction. It brings the social and environmental aspect right into the heart of pathology.
It does. You can't just look at the bug. You have to look at the context, the person, the society.
Okay, next term. Pathogenesis.
This is my favorite word on this list.
Why is that?
Because if etiology is who did it, pathogenesis is how the crime was committed. It is the mechanism. It is the step-by-step sequence of events from the moment the agent enters the body to the first cell reacting to the tissue damage, all the way to the ultimate expression of the disease. It's the story of the illness.
I like that. The story. Third term. Lesion.
A lesion is the footprint of the crime. It is the structural or functional abnormality.
So tumor is a lesion. A cut on your skin is a lesion.
Yes, but a lesion can also be invisible to the naked eye. It can be a biochemical defect in a single molecule. It is the specific thing that is physically wrong.
And finally, prognosis.
The forecast. Based on the etiology, the pathogenesis, and the lesion. What is going to happen? Will you recover? And crucially, will there be sequelae?
Sequelae, that's a good Scrabble word.
It is. It means the secondary consequences, the domino effect. A primary tumor in the lung is the disease. That tumor metastasizing to the liver is a sequela. The scar tissue that forms in the heart after a heart attack is a sequela. It's what comes after.
Okay. We have our philosophy. We have our vocabulary. Now we put on the lab coats. We are going inside the cell. Section 2. The blueprint. The genome.
And this is where we turn heavily to Robbins. Because our view of the nucleus, the cell's control center, has changed radically in the last 20 years. We used to have this very static view.
Like a library.
Exactly. The genome was a library of blueprints. You go in, you copy a blueprint for a protein, you go out and build it. Done. Very simple.
And the source material drops a number here that actually shocked me. I had to read it twice. The protein encoding genes, the actual blueprints, make up only about 1.5% of the human genome.
1.5%. It's humbling, isn't it?
It's baffling. What is the other 98.5% doing? Is it just packing peanuts, evolutionary junk?
For a long time, we arrogantly called it junk DNA. We really thought it was just evolutionary debris. But now we know it is the dark matter of the genome.
Wow.
It is regulatory.
Regulatory how?
Well, think of the 1.5%, the protein coding genes, as the actors in a play. The other 98.5%, that's the director, the stage manager, the lighting crew, and the script editor. It's filled with things called promoters and enhancers. It tells the genes when to turn on, how loudly to speak, and when to shut up.
So the complexity of a human being isn't in the number of parts we have.
No, not at all. A microscopic worm called C. elegans has about 20,000 genes. Humans have about 20,000 genes.
We have the same size parse list as a worm.
Roughly, yes. The difference, the vast complexity that makes us human, is entirely in the management instructions. That massive amount of non-coding DNA controlling everything.
Which leads us to epigenetics. This is a buzzword we hear all the time, but Robbins defines it so well as the software running on the hardware.
That's the best way to visualize it. I mean, here's the central mystery of biology. Every single cell in your body has the exact same DNA. Your liver cell has the instructions to be a brain cell. Your skin cell has the instructions to be a heart cell.
So why don't you have hair growing on your heart? Why doesn't your liver try to think?
Because the cell is only reading the relevant chapters of the book. Epigenetics is the system of bookmarks and sticky notes that tells the cell which pages to read and which to ignore. And Robbins explains the physical mechanism. It's all about histone organization.
Okay, let's unpack that. DNA isn't just floating around in a soup inside the nucleus, right?
No, absolutely not. You have about 6 feet of DNA that has to be packed into a nucleus that is about 5 microns wide. That's an incredible packaging feat.
How does it work?
It's wrapped around these protein spools called histones. This wrapped unit of DNA in histone is called a nucleosome. Now imagine a long, long string of these spools.
Okay, I'm picturing a string of pearls.
Perfect. Now, if those pearls are packed together, incredibly tightly smashed together in a dense coil, the reading machinery, the transcription factors, simply cannot get in. Physically, they are blocked.
Yeah.
This is called heterochromatin. It's dense, it's inactive, it's zipped up.
The files are locked away.
Exactly. And the open stuff.
Must be euchromatin.
Euchromatin. The pearls are spaced out. The DNA is exposed. It's accessible, ready to be read. So in one of your liver cells, the genes for make liver enzymes are in the euchromatin open for business. The genes for make a neuron are packed away in the heterochromatin, locked away, and silenced.
So pathology disease can happen even if the DNA code itself is perfect.
Precisely. This is a massive concept. You could have a perfectly normal gene sequence, no mutations. But if the epigenetic system makes a mistake and locks away a gene that should be open, say, a critical tumor suppressor gene, you get cancer.
Wow.
Or if it opens a gene that should be locked,
Yeah.
you get chaos.
The source material briefly touches on gene editing here, just as a nod to where this is all going.
Yeah, we have to mention CRISPR-Cas9. This is the frontier. Because for the first time in history, we aren't just observing these genetic and epigenetic errors, we are learning how to fix them.
And what's so fascinating is that we borrowed this tool from bacteria.
It's their immune system. Bacteria have a system to remember the DNA of viruses that attack them, and if that virus shows up again, they use this system to find the viral DNA and cut it to pieces.
A molecular scissor.
Exactly. And we've repurposed that molecular scissor. We can now, in theory, go into the human genome, find a specific typo, and precisely cut it out or replace it. It is genetic surgery.
Let's move out of the nucleus now. We're stepping onto the factory floor, section 3. Cellular housekeeping. And we have to start with the walls, the plasma membrane.
Right, and I feel like we often overlook the membrane. We think of it as just the skin of the cell, the bag that holds the water in.
But Robbins describes it as a war zone.
It is a gatekeeper. It is in a constant battle of protection versus acquisition. It has to keep the toxins and viruses out, but it has to let the glucose and amino acids in. And it's not just a wall with passive holes in it.
Right. For something small like oxygen, it can just diffuse across. But for the big, important stuff.
For the big stuff, you need active, controlled processes. One of the most important is receptor-mediated endocytosis.
That is a mouthful. Break that down.
Okay, think of it as a VIP entrance with a very specific bouncer. The cell puts out specific receptors, like millions of tiny catcher's nits on its surface. They're designed to catch one specific thing. Maybe it's LDL cholesterol.
The bad cholesterol.
Right. When an LDL particle lands in the mitt, the membrane doesn't just open a hole. It invaginates. It wraps around the molecule in the receptor and pulls the whole package inside in a little bubble.
Like a mouth closing over food.
Exactly. And this happens in these specialized reinforced areas of the membrane called clathrin-coated pits. It is an incredibly selective and controlled way to bring things in.
Now, there was a detail in here about the inside-out lipid distribution that I found fascinating. It sounded like a secret handshake or a signal flare.
It's a signal from the grave, or from the dying at least. So the membrane is a bilayer of lipids. Usually a specific lipid called phosphatidylserine is strictly kept on the inside face of the membrane. It's facing the cytoplasm.
It's indoor furniture. It's not supposed to be on the lawn.
Perfect analogy. But if a cell is dying, specifically, if it's undergoing controlled suicide, apoptosis and enzyme gets activated that flips that lipid to the outside of the cell.
So the indoor furniture is suddenly on the lawn. Why?
It's an eat me signal. It's a flag that tells the immune system's garbage collectors, the macrophages, I am dying, I am broken, come and eat me before I burst and spill my toxic guts everywhere. It ensures a clean, tidy death.
That is amazingly organized. Even in death, the cell has a protocol. Now let's talk about the skeleton, the cytoskeleton.
The cell isn't a water balloon. It's not just a sack of fluid. It has an intricate internal scaffolding. And this scaffolding is not only crucial for shape and movement, it's also a critical tool for diagnosing cancer.
There are three main types.
Three main types, each with a distinct job.
First up, actin.
Actin microfilaments. These are the muscles of the cell. They are highly dynamic. They're constantly being built on one end and disassembled on the other, a process called treadmilling. This allows the cell to move, to crawl, to change shape.
Okay, so actin is for movement. Then we have the intermediate filaments. The text calls these the ropes.
Yes, their job is tensile strength, resisting mechanical stress. But here is the aha moment for the listener. This is why your pathologist matters so much. Intermediate filaments are tissue-specific.
Meaning a muscle cell has different ropes than a skin cell.
Exactly. Muscle cells use an intermediate filament called desmin. Epithelial cells, like your skin or the lining of your gut, use a family of filaments called cytokeratins. Connective tissue cells use one called vimentin.
Why does that matter to a doctor looking at a tumor?
Okay, imagine a pathologist gets a biopsy of a tumor. The cells are so mutated, so ugly and undifferentiated that they just look like generic angry blobs. We don't know where this tumor came from. Is it a muscle cancer? A skin cancer? A connective tissue cancer? The treatment for each is totally different.
So you're lost.
You're lost. So you ask the ropes. We have stains antibodies that can detect these specific filaments. If the tumor cells light up with the stain for cytokeratin, we know. Aha, this is a carcinoma. It started in the epithelium. If it lights up for desmin, it's a muscle sarcoma. The skeleton serves as a molecular ID card for the cell's origin.
That is incredible. So the skeleton tells the history of the cell. And finally, the third type, the microtubules.
These are the highways, the rail network. They are rigid, hollow tubes. The cell uses them as tracks to transport organelles and vesicles from one part of the cell to another. Little motor proteins, kinesins, and dyneins literally walk along these tubes carrying cargo.
Like a little train system.
Exactly.
Yeah.
But they also have another job. They form the mitotic spindle, machinery that physically pulls the chromosomes apart during cell division.
Which makes them a prime target for chemotherapy drugs.
Right. A massive target. Drugs like paclitaxel or Taxol are a very common chemotherapy. And they work by stabilizing these microtubules, essentially freezing them. If the cancer cell can't build and break down the scaffold to pull its chromosomes apart, it can't divide. It gets stuck in mitosis and dies.
We can't talk about a house without talking about the trash removal system, the janitors of the cell, lysosomes and proteasomes.
A cell produces a huge amount of waste. Misfolded proteins, old, worn-out organelles, bacteria it might have swallowed. If it doesn't clean up after itself, it dies. The lysosomes are the stomach of the cell. They are acidic bags full of powerful digestive enzymes.
And this is where autophagy comes in.
Autophagy. Self-eating. Sounds morbid, but it's a critical survival strategy.
Yeah.
Let's say a cell's starving.
Right.
Say you are fasting and glucose levels are low. The cell needs fuel and building blocks.
So it looks around the house for things to pawn.
It does. It will look at its own internal furniture, maybe some old mitochondria that aren't working great anymore, and will wrap them up in a membrane and feed them to the lysosome.
It cannibalizes itself to survive.
It recycles the parts to keep the vital functions running. It's incredibly efficient.
And the proteasomes, what's their job?
They are the shredders, but they are more specific. They handle individual proteins. If a protein is misfolded or damaged, which happens constantly, the cell tags it with a small molecule called ubiquitin,
The source material calls it the trash tag.
or the kiss of death. The proteasome is a barrel-shaped machine that recognizes this ubiquitin tag. It grabs the tagged protein, sucks it in, and shreds it into its constituent amino acids, which can then be recycled.
So what happens if this system fails? If the shredder gets jammed?
Junk protein builds up. It accumulates, and this is bad news.
Why?
These misfolded proteins are sticky. They start to clump together, forming aggregates. And these aggregates are toxic. They disrupt cellular function. This is the fundamental basis of many neurodegenerative diseases.
Like Alzheimer's.
Alzheimer's, with its amyloid plaques and tau tangles, Parkinson's with its Lewy bodies. These are all, at their core, diseases of aggregates. They're diseases of protein junk piling up because the janitorial staff couldn't keep up with the mess.
This transitions perfectly to Section 4, energy and decision-making. The mitochondria.
And everyone together now. the powerhouse of the cell.
It's the meme. It's the one thing everyone remembers from ninth grade biology. But Robbins paints a much, much darker picture. They aren't just the power plant, they are also the executioner.
This is one of the most important dualities in all of biology. Yes, mitochondria perform oxidative phosphorylation. They burn glucose with oxygen to make massive amounts of ATP, the cell's energy currency. They keep the lights on, but they also hold the keys to life and death. Hidden away, tucked inside the inner membrane of the mitochondria, is a protein called cytochrome C.
And it's supposed to be in there, right?
It's an essential part of the energy-making machinery inside the mitochondrion. But if the cell is stressed beyond repair, if its DNA is fried from radiation, or if the load of misfolded proteins is too high, the cell makes a decision. It decides it's time to die.
And how do the mitochondria execute that order?
The cell triggers the formation of pores, of holes, in the outer mitochondrial membrane.
And the cytochrome C leaks out?
It leaks out into the cytoplasm. And when cytochrome C hits the cytoplasm, it acts like a poison pill. It triggers a deadly cascade of enzymes called caspases that act like molecular guillotines, systematically chopping up the cell's DNA and proteins from the inside out.
So the mitochondria are holding a grenade, waiting for the order to pull the pin.
It is a profound concept. The thing that gives you life is also the thing that kills you. It's a fail-safe. You want damaged cells to die. You don't want them sticking around, malfunctioning, and potentially becoming cancerous.
Now, there's another mitochondrial concept here that is absolutely crucial for understanding cancer, the Warburg effect. This one always confused me, so walk me through it slowly.
It confuses a lot of students, so you're not alone. Here's the setup. Normally, your cells burn glucose using oxidative phosphorylation because it is incredibly efficient. You get about 36 ATP molecules per molecule of glucose.
A huge energy payout.
Huge. But cancer cells and other rapidly dividing cells do something weird. Even when there's plenty of oxygen available, they switch to a different, much less efficient method, aerobic glycolysis. This only gets you 2 ATP per glucose.
Which seems completely backward. Why would a rapidly growing cancer cell, which needs tons of energy, choose a terrible fuel source?
Because energy isn't the only thing a rapidly growing cell needs. Think about a construction site. If you're building a new skyscraper, do you just need electricity to run the tools?
No, I need steel, I need concrete, glass, pipes.
Exactly, you need raw materials, building blocks. By running this inefficient glycolysis pathway, the cancer cell generates a ton of metabolic intermediates, carbon chains, that it can siphon off to build new membranes, new DNA, new amino acids.
So the Warburg effect is about sacrificing energy efficiency to get more building blocks.
Exactly. It's a strategic supply chain shift. The cancer cell isn't just trying to power itself. It's trying to build a whole new cell. It's not broken. It's repurposed for growth.
We've used the word suicide a few times now. We need to clearly distinguish between the two ways a cell can die. Necrosis versus apoptosis.
This is a fundamental distinction. It's the difference between a car crash and a quiet, planned retirement party.
Okay, explain necrosis.
Necrosis is the car crash. It's messy, uncontrolled, accidental death. Let's say you have a heart attack. Blood flow to a part of the heart muscle stops. No oxygen.
Okay.
The mitochondria fail. No more ATP. The pumps on the cell membrane that keep water and ions balanced, they stop working. Water rushes in, the cell swells up like a balloon, the membrane bursts, and the cell spills its guts into the tissue.
It's messy.
It's incredibly messy. That spillage of intracellular contents causes a massive inflammatory response. It calls in the immune system, which causes even more collateral damage to the neighboring cells. Necrosis is always pathological. It's always a result of injury.
And apoptosis.
That's the retirement party. It is programmed, controlled, clean cell death. The cell realizes it's time to go. Maybe it's old, maybe its DNA is damaged beyond repair, maybe it's just not needed anymore, like the webbing between your fingers and toes when you were an embryo.
So how does it go out?
Quietly. It shrinks down, its nucleus condenses, it packages its contents into neat little membrane-bound parcels called apoptotic bodies. It puts out that eat me signal, the phosphatidylserine we talked about earlier. A macrophage comes along, gobbles it up, that's it.
No inflammation, no mess.
No mess. It leaves the tissue exactly as it found it, just with one less cell. It's a vital process for normal development and tissue maintenance.
Okay, let's move on to section 5, communication. As they say, no cell is an island.
If a cell stops communicating, it's usually very bad news. It either dies of loneliness or it goes rogue and becomes cancer. Cells have to constantly talk to each other to maintain tissue order.
And there are a few ways they do that.
Right. You have paracrine signaling, which is basically talking to your immediate neighbors.
Yeah.
Endocrine is long-distance calling using hormones in the bloodstream.
But I want to focus on the extracellular matrix, or ECM. I used to think this was just the stuff between cells, like the mortar between bricks.
Yep.
It's dead filler space.
That's the old view. We now know the ECM is an incredibly active and complex environment. It's not mortar, it's more like soil. It provides not just structure, but also signals that regulate whether the cell grows, survives, or dies.
The outline mentions the basement membrane.
That's a specialized sheet-like layer of ECM. It's the floor that all epithelial cells sit on. But it's not just a floor. It provides orientation. It tells the cell which way is up and which way is down, a concept called polarity.
And how do the cells know they are attached? How do they feel the floor beneath them?
Through proteins called integrins. These are fascinating. They are transmembrane proteins. So imagine a protein that sticks all the way through the cell membrane. On the outside, it has a hand that grabs onto the ECM proteins like collagen or laminin. On the inside, it has another hand that connects to the cell's internal cytoskeleton.
So it's a physical, mechanical link from the outside world to the inside skeleton.
Yes. It's how a cell feels its environment. A cell can sense the stiffness of the matrix it's on through these integrins. And here's the kicker. For most normal epithelial cells, if their integrins let go of the ECM, if they become detached, the cell panics.
It feels like it's falling, untethered.
In a way, yes. It triggers apoptosis, it initiates a self-destruct sequence, it knows it is lost, and is a danger to the organism. This specific type of apoptosis is called anoikis, which is literally Greek for homelessness.
Homelessness-induced death.
Right. So think about what a cancer cell has to do to metastasize. It has to break off from a tumor, float through the blood or lymph where it's homeless, and survive long enough to land somewhere else. It has to figure out how to hack the system and overcome anoikis.
Finally, let's talk about the future of the tissue. Section 6. Growth, renewal, and stem cells.
It all comes down to the cell cycle, the clock of cell division. G1, S, G2, M growth, synthesis of DNA, more growth, and then mitosis. But the most important parts for pathology are the checkpoints.
The quality control stations.
Exactly. There is a major checkpoint at the transition from G1 to S phase. Before the cell commits to the massive undertaking of copying its entire genome, it stops and asks, is my DNA healthy? Is the environment favorable for division?
And if the DNA is damaged, if there's a mutation or a break?
A protein called P53 steps in.
P53. This is probably the most famous molecule in cancer biology.
It is the guardian of the genome. That's not an exaggeration. If P53 detects DNA damage, it halts the cycle. It slams on the brakes. It gives the cell time to repair the damage.
And if the repair works?
If the repair crew fixes the DNA, P53 gives the all clear, and the cycle continues. But if the repair fails, if the damage is too severe, P53 makes the ultimate call. It triggers apoptosis. It executes the cell to save the organism from propagating mutations.
And if P53 itself is mutated?
That is the nightmare scenario. If the guardian is asleep or broken, the brakes are cut. The cell with damaged DNA divides anyway. The mutations are passed on to the daughter cells, and those daughter cells divide, accumulating even more mutations. This is why P53 mutations are found in almost 50% of all human cancers. The guardian is asleep at the wheel.
Let's wrap up this section with stem cells. We hear about them constantly in the news, stem cell therapy. But biologically, what is the defining feature?
Two things define a stem cell: the ability to self-renew and the ability to differentiate into specialized cells. But the key mechanism, the magic trick that allows this, is asymmetric division.
What does that look like?
Okay, when a normal skin cell divides, it makes two identical skin cells. When a stem cell divides, it does something different. One daughter cell becomes a specialized worker. It goes off to become a skin cell or a gut lining cell. But the other daughter cell, it remains a stem cell.
So you spend one, but you keep one.
Exactly. You maintain the pool. You don't deplete your reserve of stem cells. And these precious cells live in special, protected microenvironments called niches.
Like the VIP lounge.
Or a bunker.
What?
Deep in the crypts of the intestine, or deep in the bone marrow. The niche provides signals that protect the stem cells from damage and keep them quiet until they are needed for tissue repair. Because if your stem cells get corrupted, the entire tissue is doomed.
This has been a whirlwind tour of the microscopic. I mean, we've gone from the philosophy of normality to the mechanics of the cytoskeleton to the suicide logic of mitochondria.
And this brings us all the way back to our mission. Why does this matter? Why should a non-pathologist care about integrins or P53?
Because every single disease starts here.
Every single one.
Huh.
A heart attack isn't just the heart stopping. It is ischemia leading to mitochondrial failure, leading to ATP depletion, leading to membrane pump failure, cellular swelling, and ultimately necrosis. It is a story written at the cellular level.
And cancer isn't just a lump.
It's a story of a failure of DNA repair, a silencing of tumor suppressors via epigenetics, a hijacking of the cell cycle checkpoints, a hacking of the Warburg metabolism, and a manipulation of the ECM to allow for invasion. It is a disease of this very machinery we've been discussing.
This synthesis of Robbins and Underwood really highlights that pathology is probabilistic. It's all about resilience and reserve.
That's the final thought I really want to leave with you. Our cells are incredibly resilient. They have backup systems, repair crews, janitors, and fail-safes. Disease only happens when that reserve capacity is exhausted, when the stress is too great or lasts too long and adaptation finally fails.
So to our listeners, take this mental map, use it. Go and read the chapter on the cell in Robbins. Read the chapter, What is Disease in Underwood. You will see these concepts on the page, and hopefully they will click into place because you know the characters in the story now.
And maybe think about this as you go about your day. You are a galaxy of trillions of these little units. All of them are communicating. All of them are making energy. All of them are constantly balancing on that tightrope between successful adaptation and catastrophic failure. It's not just lucky that we are healthy. It is an ongoing miracle of organization.
That is a provocative thought to end on. If we understand the cell, we understand the suffering, but we also understand the miracle of life itself. Thanks for diving deep with us.
My pleasure.
We'll see you next time.